Welcome to today's Advanced Modeling module. In this Advanced Modeling lecture, we'll be talking about an agent-based model. My name is Yunjin, and I'm a PhD student at Cornell University, and I'll be guiding you through the basics and applications of an agent-based model throughout this lecture today. So we will be dividing today's lecture into 4 parts. First, we will learn about what an agent-based model is, which I'll refer to as ABM throughout this lecture. Second, we will explore some applications of agent-based model in produce safety. Third, we will learn about how we can use the results from ABM to improve produce safety outcomes. And finally, we will compare ABM with other modeling tools that are used in produce safety applications and learn when it is better to use than the others. At the end, I will suggest some resources that you can refer to if you want to learn more about ABM applications in produce safety. After watching today's lecture, you will be able to understand what ABM is and explain the key components of ABM, along with the characteristics of ABM. You will also be able to describe the steps to evaluate the data generated from ABM, understand what kind of data can be collected and how they can be used, interpret the results from statistical analysis performed on the data generated from ABM. You'll also be able to communicate the findings from the analysis to support the decision-making process in produce safety. And finally, you'll also be able to evaluate where ABM can be applied in produce safety and compare ABM with other modeling tools. So let's dive into the first part of our lecture. What is an agent-based model? So ABM is a computational model that simulates the interaction between agents within a virtual environment. ABM can replicate a system in reality to conduct a virtual experiment and simulate consequences of agent interactions over a period of time. To explain ABM in simpler terms, let's say that we're modeling a traffic flow. Each car would represent an agent, and each car can slow down, speed up, or change lanes based on the behavior of the other cars. By simulating this model, we can study different driving behaviors or how each car can affect the overall traffic flow and how traffic jams are created. Let's get into learning more about different components of ABM. Here, I'll use an example of a floor map of the produce section of the retail store that was developed in NetLogo to explain further. Note that although NetLogo is a commonly used software for ABM for its user-friendly and intuitive graphical interface, ABM can be developed in any programming language such as R, but for this lecture, I'll be explaining the applications and examples based on an ABM developed on NetLogo. Okay, so in an ABM, there is an environment, which is a space where agents show their behaviors. This example here shows a floor map of where things that could be involved in pathogen transmission are located in the produce section of the retail store. This area here represents the storage room. This area represents the cutting station. This area shows different dry produce display shelves, and this specific area represents the misted wet shelf section. The dots, triangles, and other icons like human or cart you see on this map represent each agent, which I'll explain further soon. Although the environment here is specific to the produce section of the retail store, any environment in a produce supply chain, such as a processing facility or a packing house, can also be replicated into NetLogo. So what is an agent? Agent is an individual or an entity within the system. From our example, an agent would be any environmental surfaces that could be involved in the pathogen contamination or transmission. The agents on this map are represented as dots, triangles, a human figure, or cart. Let's zoom into each of these a bit. Here, you see a human figure and a cart figure. The human figure here would represent an employee that specifically works in the storage area, and the cart figure would represent a runner cart that an employee used to distribute produce across the produce section of their retail store. In this part of the map, where I mentioned was a cutting station, the triangles represent a hand wash sink and the handle of a refrigerator. The dots from the main produce display section represent each dry produce display shelf. And as you can see on that logo, these agents are connected via lines, which represent their interactions. In an ABM, we can model an interaction between the agents or the environment based on the rules. Let's go back to this interaction where we see this line between human figure and a cart. Here, we're simulating an interaction between the employee and the produce cart, where the employee is transferring the pathogen to the handle of the cart as it comes with a contact. This is where model parameters that we are initially inputting to build the model are used, as we use these numbers to simulate things like what are the chances that the employee would transfer the pathogen onto the cart and how much the pathogen would be transferred if the transfer occurs. During each of these interactions between all the connected agents, each agent follows a set of rules. The rules are defined methods on how agents behave. For example, the ABM of a produce section of the retail store shown here has different events scheduled during a simulation. A part of this map shows a cutting station where the produce are pre-cut and the agents in this location get cleaned every 4 hours during the store operation. Meanwhile, the agents in other locations like dry display shelves get cleaned less frequently than the agents in the cutting station. So different rules can be applied to different agents. Therefore, ABM allows us to simulate the behaviors of all these different components that could go into the model simultaneously.
Based on what we just discussed about the components of ABM is unique in that it is autonomous, which means that agents can follow a set of rules independently. It also has heterogeneity, meaning that each agent has different characteristics, such as different hygienic zone level, different type of surface materials, which will allow the modeler to integrate different parameter values specific to an agent. ABM is also discrete in a way that different events can be accounted for simultaneously in the model. For example, pathogen introduction from incoming produce can occur while pathogen introduction from consumer to produce display shelves also occurs. And again, ABM is dynamic as it can account for interaction between agents. So let's move on to part 2 of our lecture. What are some of the applications of ABM? Although we will be focusing on how ABM can be used in produce safety context, ABM has been widely used across different disciplines. In biology, a common simulation model that has been used is a predator and prey model on how each population change over time. In urban landscapes, ABM has been used to observe the ecological impact of cities on the environment to evaluate the land use policies. And in economics, ABM has been used to simulate stock market. ABM has also been used in geology to model the spread of wildfire. So let's take spread of wildfire as an example to see how ABM has been used in other fields. The figure here shows an example provided by Uri Walensky developed on NetLogo. Here, the environment is replicated as a grid of cells, which forms a map on NetLogo, and some of these cells are represented as trees. Certain cells that are on fire are assigned with a set of rules, and the wildfire is simulated to see how it spread to neighboring trees. The researchers can use this to experiment how different densities of trees in the forest can affect the wildfire spread, and it can be modified to account for different environmental factors, such as wind speed and type of wood.
Now that we saw how ABM can be applied in different fields, let's learn how ABM can be applied in produce safety. ABM can be mainly used to simulate potential routes of pathogen contamination and transmission within an environment across different parts of the produce supply chain. For produce fields on a pre-harvest level, ABM can be helpful when identifying high-risk areas to collect pre-harvest samples or inform setback distances for produce harvesting in the event of flood. If we were to simulate produce processing facilities or packing houses, ABM can be also used to identify high-risk areas for collecting environmental samples, and also to suggest targeted corrective actions and pathogen control strategies. ABM can be even applied towards the end of the produce supply chain, which is the produce section of the retail stores, to identify high-risk behaviors and pathogen contamination pathways within the retail environment and inform how pathogen contamination level might change at different levels of consumer traffic. Moving on to part 3 of our lecture, we're going to learn how we can use the results from ABM to improve produce safety outcomes. When you're developing an agent-based model, it is good to think of what your outcome of interest is that suits your objective. In a produce safety application, the overall pathogen level or prevalence of the environment would be the major outcome of interest. Other risk attributes, such as the number of times pathogen is transferred to other agents or the total time the pathogen is detected on an agent, could be other outcomes that could be collected. Then, you want to decide how long you would like to run the model. It can be several months to years, and it is really up to you, but it will be important to decide at which time points you would like to collect the data. Let's say your overall simulation would represent a year in reality, and you want to collect the pathogen level at the end of each month or every 3 months. Or you might just want to aggregate the data at the end of the simulation. So it is really up to the modeler to decide what kind of data and how much of the data you would like to collect. Using the synthetic data that you collected from ABM, you can run different statistical analysis to support the decision-making process in produce safety. While there are numerous statistical analyses that can be used to analyze the output from ABM, I want to introduce some of the analysis that could be particularly useful in a produce safety application. The 3 analyses that we will go over in our lecture are sensitivity analysis, cluster analysis, and what-if scenario analysis. In the following slides, we will be learning about what these analyses are and how the results from these analyses can be used to aid with the decision-making process in produce safety. I'll be using specific statistical analysis results from the agent-based model developed on the produce section of the retail store, which you also saw the map of in the beginning of this lecture. And I'll be using this to explain the application of these analyses and how the results have supported some of the decision-making process to improve overall produce safety. First, we will learn about sensitivity analysis. Sensitivity analysis help us to identify the parameters that affects the outcome of interest. In the case for the ABM developed based on the produce section of the retail store, the major outcome of interest were the prevalence and concentration of Listeria. So using the sensitivity analysis, we will be evaluating how much each model parameter, such as transfer coefficient between agents and Listeria prevalence or concentration on incoming produce, affect the overall Listeria prevalence and concentration in the produce section of the retail store. Although there are different types of sensitivity analysis methods that can be used, in this lecture, we'll be going over partial rank correlation coefficient, also known as PRCC method. So this method is used to evaluate nonlinear monotonic relationship between variables. Overall, conducting sensitivity analysis can help identify high-risk factors and suggest better characterization of sensitive parameters in future model development. And it can also allow the modelers to adjust the parameters for what-if scenario analysis later to evaluate different control strategies. Here we see 2 figures showing the sensitivity analysis results that used partial rank correlation coefficient method. The left hand of each figure shows the abbreviations of all the model parameters that were evaluated in the ABM developed for the produce section of the retail store, which I won't go into too much details, but the parameters highlighted with red asterisks show the top 5 sensitive parameters that were statistically significant. Let's first look at the top 5 parameters on the left figure. These top 5 parameters identified to be most sensitive to overall Listeria prevalence at the retail store were initial Listeria concentration on incoming produce, transfer coefficient from incoming produce to employee, since employee are the ones who have the first contact with the incoming produce, transfer coefficient from consumer to produce display shelves, probability of transfer from hygienic zone 1 agent to other zone 1 agents, and the number of total maintenance events that occur at the retail store. The same parameters were identified as top parameters affecting the overall Listeria concentration on the right figure in the produce section of the retail store. However, some of them had some different orders, but the top 2 parameters still remain the same. So these results suggest that obtaining accurate Listeria concentration on incoming produce and transfer coefficient from incoming produce to employee are important, as they can drive the overall Listeria prevalence and concentration at the produce section of the retail store, and also allow us to develop targeted corrective actions that could be tested with what-if scenario analysis, which I'll explain further later.
Another statistical analysis that we will learn about is cluster analysis. Cluster analysis groups similar agents with distinct contamination patterns that can help with optimizing an environmental sampling plan. There are multiple different types of cluster analysis, but the one that I would like to introduce that can be useful in a produce safety application is hierarchical cluster analysis. For this analysis, the distance that measures the similarity between every data points, in our case, agents, is measured for each risk attribute that we are interested in. And specifically, for an agglomerative method, which is also known as a bottom-up method, adds one data object at a time and merge them to form a bigger cluster until one big cluster is formed. You can choose any risk attributes that you would like to include to group the agents based on the contamination patterns. And some examples of risk attributes that you can collect are total time pathogen is detected on an agent throughout the simulation, total number of times that the agent transfers the pathogen to other agents, mean pathogen prevalence and concentration, or even rare contamination events like the number of roof leak events or maintenance events that introduce pathogen to an agent. The continuous risk attributes listed here were all the risk attributes that were included for the cluster analysis conducted for the agent-based model developed for the produce section of the retail store. The figure here shows the dendrogram created as a result of a cluster analysis that was conducted using R. The height of the branches show how similar or dissimilar clusters are, so the longer the branch shows that the clusters are more different from one another. The dendrogram here shows that all the agents identified in the retail store ABM were grouped into 6 unique clusters. I won't be going through this in great detail, but you can see that some clusters have a wide variety of agents, while other cluster has relatively uniform agents in a cluster. Each risk attribute for each cluster were summarized to identify their distinct contamination patterns. Since some of the risk attributes are like mean Listeria prevalence or concentration, cluster analysis can inform us which sites are at higher risk to focus the sampling on, and also reduce cost in sampling, as you can also reduce the number of sampling for similar sites that share similar contamination patterns.
The last example of statistical analysis that I want to introduce here is what-if scenario analysis. The goal of the what-if scenario analysis is to test various scenarios and provide insights on predicted impacts on our outcome of interest, which in our case would be pathogen prevalence and contamination level. Your scenarios can be different corrective actions based on some of the sensitive parameters identified from sensitivity analysis, or changes in cleaning schedules or cleaning frequency that could change the pathogen spread patterns. So it is good to think of feasibility of these scenarios and how they can be applied in reality as you come up with these scenarios. Now, let's look at some examples. So we'll be going over some of the scenarios that were implemented in the ABM that were developed for the produce section of the retail store, but similar scenarios can also be applied in different parts of the produce supply chain, like in a processing facility or a packing house. The first what-if scenario that was tested was reducing Listeria prevalence on incoming produce. So in reality, this could be done through a more stringent supplier qualification. To implement this scenario in the model, the initial distribution of Listeria on incoming produce were reduced by 50% and 90% to see the effect on overall Listeria prevalence and concentration in the retail store. If one of the scenarios that we can evaluate is to reduce the introduction of pathogen into the environment, another scenario that we can evaluate is change in cleaning schedules or cleaning efficacy. For one of the scenarios, cleaning and sanitation frequency was increased on produce display shelves. But we can also target a specific type of agent, such as the scales that consumers use in the produce section, which are not usually cleaned as often or does not have a regular cleaning implemented, to see how it might affect the overall pathogen level in the retail store. Finally, other than corrective action scenarios, we can also account for special occasions such as holidays in the case of a retail store. Let's say we are trying to see if the month that has a holiday has any change in overall Listeria contamination level at the retail store. We can implement this by changing the operation schedule that accounts for the changes in the retail store, changing traffic level 1 or 2 days or several days before the holiday. So for example, if we were to simulate a month that has a Thanksgiving Day, usually there will be more consumers trying to buy ingredients to cook for the holiday. And since the employees working on that day at the retail store will also have a shorter day, there could be a decrease in cleaning frequency, or third-party deep cleaning events might be canceled. So ABM can account for all these changes to see how the agents might interact differently, or how pathogen transmission might change over time simultaneously. So it could be really useful when trying to simulate a real-life scenario. So based on the scenarios developed, we would have compared how much of a change in Listeria prevalence and concentration had for each scenario with the baseline model result. Based on how much Listeria has been reduced, we can identify how effective each corrective action is and suggest some of the corrective actions that can take place in reality. For example, the what-if scenario analysis for the retail ABM showed that controlling Listeria from incoming sources, especially from the incoming produce, is the most effective strategy to control Listeria in a retail environment. This can suggest that stringent supplier qualification would be a key to controlling Listeria at retail. And another effective corrective action that could be done at the retail store is increasing deep cleaning frequency and verifying cleaning efficacy. So even if retail store has regular cleaning events, there are sections that do not get cleaned as frequently as other sections, such as the dry display shelves would not get cleaned as often as the wet display shelves. So targeting on the sections that get infrequent cleaning events can reduce overall Listeria level, and verifying that the cleaning was actually efficiently done would be very important. So these results overall suggest the retailers where to focus on when they are developing Listeria control strategies, and use the model to simulate targeted corrective actions before they implement in reality. Now that we went through the basics of how each analysis works and how they can be applied, I want to take a second to explain how these statistical analysis results using the data generated from ABM could be useful in improving produce safety. One of the key aspects of controlling pathogen involves an environmental monitoring program that regularly collects samples from the environment to identify which sites are at risk for contamination. So ABM can be very useful in optimizing pathogen environmental monitoring program, and each analysis can play a role. For sensitivity analysis, it can help identify the biggest risk factors of pathogen contamination and transmission in a given environment. Cluster analysis can group the sites with similar contamination patterns based on chosen risk attributes to reduce the number of sampling, to reduce sampling cost on lower risk areas and focus on the higher risk areas. And what if scenario analysis can simulate various corrective action scenarios to evaluate their efficacy and develop targeted control strategies specific to the environment. So now moving on to the last part of our lecture, let's explore when ABM can be better than other modeling tools. First, let's see what some of the advantages of ABM are in produce safety. As previously mentioned when we were talking about the characteristics of ABM, ABM has a dynamic nature because it can simulate complex behaviors and interaction between individuals, which is not commonly seen in other types of models. It is also flexible due to its heterogeneity that can be considered among agents and their dynamic behaviors.
Then what are some of the limitations of ABM? The downside of using an ABM is that it is not too generalizable. A modeler has to build a model specific to the environment, so it cannot be easily replicated. If you have developed a model for a retail store, you will not be able to use the exact same model for a processing facility. If you have a previously developed ABM, you can still adopt similar features or use some of the model parameters, but you will still have to tailor it to fit the specific environment you are trying to model. So developing an ABM can be time-intensive and computationally intensive. Next, there's limited data available out there to use as model parameters. Although you can use the data available on literature as model parameters, it is hard to find the exact values that fits to the specific facility that you're trying to model due to some of the practical challenges. And finally, there is no standard method for validating the ABM. Although researchers have been using the most reasonable methods and data to perform the validation process, similar to other simulation models, validation is still a challenge for ABM. So this would require further research to develop a standard validation method.
In addition to ABM that we introduced in this lecture, there are other modeling tools that are useful in produce safety application. Quantitative microbial risk assessment, also well known as QMRA, and geographic information systems, GIS in short, are the two that we are going to compare with. We also have a modeling lecture prepared for both of these models in our module. If you're interested in learning about QMRA, you can refer to lecture 2 of our undergraduate modeling module, and you can watch the other lecture in the Advanced Modeling Principle series if you want to learn more about GIS.
Okay, so let's take some time to compare an agent-based model with QMRA and GIS to help you decide when to use which modeling tool. We just went over the advantages and limitation of ABM, but just to briefly reiterate them for the comparison. Some of the strengths of ABM are that it can simulate complex behaviors and interaction between individuals, and it is flexible due to heterogeneity and dynamic behaviors. However, ABM can be computationally intensive and difficult to validate, and it requires very specific data to design the system. But ABM can be better than other modeling tools when you are trying to model discrete events and simulate a complex system. Let's now look at QMRA. There have been a lot of QMRA developed in produce safety applications, so it is very well established and standardized. Also,
QMRA focuses on the entire supply chain from production to consumption instead of the specific stage in a supply chain like ABM. However, the model oversimplifies the complex system, so you cannot incorporate like dynamic behaviors as well as ABM. So QMRA can be a better modeling tool when you're trying to evaluate risk on a higher level, and it also has a more straightforward model development process. For GIS, the data sources for GIS are readily available and they are constantly updated. So GIS has the least issue with the accessibility with data sources compared to any other modeling tools. Therefore, it can be best used for data-driven decision support process compared to other models where some values are based on assumptions. However, GIS is mostly focused on the pre-harvest level since the environmental factors incorporated in the model are focused mostly on the pre-harvest level, so it does not provide a comprehensive overview of the food supply chain. So GIS can be definitely a better modeling tool when modeling a pre-harvest level pathogen contamination, and if you want to further develop the model as a real-time monitoring tool. Now that you have watched this lecture on the overview of ABM, there are a few things that I want you to take away from this lecture. I hope you learned that ABM can be a useful tool when simulating potential routes of pathogen contamination or transmission within an environment across different parts of the produce supply chain, and that you can utilize the results from ABM to optimize pathogen sampling plans and suggest targeted corrective actions to improve produce safety. And finally, it is important to understand the strengths and weaknesses of each modeling tool, such as ABM, QMRA, and GIS that we went over to better evaluate which tool best fits your objective. If you are interested in learning more about the background of ABM or planning to develop an ABM yourself using NetLogo, I would suggest looking into the Introduction to Agent-Based Modeling textbook by Walensky and Rand. I also want to point out that there are various agent-based models that has been developed to simulate pathogen cross-contamination in processing facility. This particular paper also uses an agent-based model framework and developed a model using R, which would also provide an insight on ABM that was not developed on NetLogo. I would also like to highlight the series of papers that developed on ABM to simulate Listeria level in different parts of the produce supply chain, all the way from fresh cut produce facility to the produce section of the retail store, which we use the results to explain the statistical analysis in this lecture. This modeling lecture was made with the support that was provided as part of the grant from Specialty Crop Research Initiative from USDA NIFA. And these are all the references to all the papers or sources that were mentioned throughout the lecture. Thank you so much for joining me to learn more about an agent-based model today, and I hope you can use this as a basis to develop your own ABM to assist with improving produce safety with me in the future.